Welcome back once again here at Aries Impact International Academy, and we're going to be continuing from our last session. We've concluded discussing the non-essential structures that we have in a bacterial cell, and now we're going to be discussing some of the essential organelles that you have in the bacterial cells. And so these essential organelles, as we highlighted earlier, they involve the cell wall. We're going to also discuss the plasma membrane, and then we'll look at the cytoplasms and the components found within the cytoplasm. Alright, quickly we'll be diving right into the cell wall. Now, the cell wall of a bacterial cell is made up basically of murines. Are you getting me now? It's made up of murines. And these murines play a vital role in determining what kind of bacterial cell we are dealing with. Because the cell wall of bacterial cells is used as a distinguishing factor to differentiate between the two major classes of bacteria that we have when you're talking about in terms of cell wall, which is what? Gram-positive bacteria and the gram-negative bacteria. But before we go there, what are murines? Murines are also they are the building block that forms what we call peptidoglycan. So these peptidoglycan are found within the cell wall of a bacteria, and they are usually made up of what we call murines. Murines are majorly modified forms of glucose that we have, and we have them NAG and NAM. And when we say NAG, we are talking about the N-acetyl glucosamine. So we have the N-acetyl glucosamine. You understand, is a murine, which is one of the components of peptidoglycan. And then you also have the NAM. And the NAM stands for N-acetic muramic acid. So we have the N-acetyl muramic acid. N-acetyl muramic acid. So the N-acetyl glucosamine and N-acetyl muramic acid is what we make up murines. There are different kinds of murines we have, and they form the peptidoglycan, which is a major component of the cell wall. Now, like I said earlier, bacteria are classified based on the nature of their cell wall. That's like saying based on how their organization, the organization of their peptidoglycan, which I said is a major component of cell wall, is. So good. Now, classifying bacteria based on cell wall, we are classifying them now based on cell wall. I said we are going to be looking at gram-positive bacteria and gram-negative bacteria. So we have the gram-positive bacteria and we have the gram-negative bacteria. Why did they use the word gram? Because the gram stain technique is the technique used in exploring their cell wall differences to be able to see the difference between those two kinds of bacteria. Alright, good. So, what does this staining technique look like? Before we describe the staining technique, I want us to appreciate this diagram here, because the diagram will lay a lot of light in understanding the staining, the principle of this gram staining technique, which they can ask you in your exams. Alright, good. These gram-positive bacteria, basically, on a very simple note, we can say that these are the bacteria that have thick peptidoglycan. So if we're talking about gram-positive, they have thick peptidoglycan. But when we're talking about gram-negative, they have thin layer of peptidoglycan. And I told you that peptidoglycan is what responds for the thickness of the cell wall. So automatically, that's like saying that gram-positive bacteria have a thick cell wall. They have a much thicker cell wall, while this one have a much thinner cell wall. Another thing again that your gram-positive bacteria also have is that they have the presence of something which we call lipoproteins. Now, these lipoproteins are of different types. You understand? And we're going to look at the first one, which we will call lipoteichoic acid. Lipoteichoic acid, and then we also have the teichoic acid. So good. We'll explore the differences and the use of all these lipoteichoic acid and teichoic acid. But basically, before I even talk about going into that exploration, one general thing I need you to know about these two things here is that both of them are made up of glycerol phosphate. Glycerol phosphate. Just in case you see it in an exam, they are made up of glycerol phosphate, and they are also made up of ribitol phosphate. In our biology 001 lectures, you would have seen where we explore glycerol phosphate and ribitol phosphate, but we're just bringing it into this concept again for you to know that this lipoteichoic acid and teichoic acid are made up of this glycerol phosphate and ribitol phosphate, and they are present in gram positive but absent in gram negative. So in gram negative, what we really have there is LPS. So in gram negative, we have what we call LPS, that's lipopolysaccharide. So when you hear LPS simply stands for lipopolysaccharide. Let me just put that here so you don't forget that. So LPS stands for lipopolysaccharide. Yes. Alright, good. So when we come to this diagram here now, this is the diagram of the cell wall. In fact, precisely, this one here is the diagram of the cell wall of gram-positive bacteria cell wall, while this one here is that of the gram-negative bacterial cell wall. Are we there now? So good. These red circles, these red oval structures here, we are going to let that represent our N-acetylmuramic acid, while the blue colored ones, let me just highlight it beside. So this is N-acetylglucosamine, and then the red one there is standing for N-acetylmuramic acid. So here now we are looking at the cell wall, and this is like the inner membrane. Okay, good. So this is the cell wall. Now, what makes up the cell wall, if you notice? If you look here and look here, you see that we have almost like how many layers? 1, 2, 3, 4, 5, 6. Like 6 layers of peptidoglycans. Are you getting me now? Unlike here that you just have just 2 layers of peptidoglycan. So which of them have thicker amount of peptidoglycan? This one right here, that's the gram-positive. So we can appreciate that when we said we have thick peptidoglycan in gram-positive bacteria. So you can see that here. So we are just going to be highlighting some things here. I just quickly want to highlight something here. So in this place here, this layer here, we can consider this layer the peptidoglycan layer. So this is the peptidoglycan layer. Peptidoglycan layer, right? Now this space in between this peptidoglycan layer and our inner membrane, this is like an inner membrane, or you just that this is the only membrane in gram positive. Is in gram negative you have inner and outer membrane. So in gram positive bacteria, another important thing you want to know is that what they lack outer membrane. So you want to remember that they lack outer membrane. They only have inner membrane. Yes. So these ones have outer membrane and also have inner membrane. So just remember that. Okay. So this one lack outer membrane, this one have inner membrane. So you can see that also clearly here. This is a membrane and this is another membrane. So this one inner membrane, outer membrane. But here you only have the inner membrane. I get it now. And in between the peptidoglycan layer and the inner membrane, you have this space here, and this space here contains components that can release enzymes and some other stuff, but they call it the periplasmic space. So it is called the periplasmic space. Yes, this periplasmic space here, you can see that it's empty, there's no peptidoglycan in it, right? But this space in gram negative is also the periplasmic space. It's the periplasmic space, and I'm sure you can begin to explore some differences between gram negative and gram positive also. So you can see that in gram negative bacteria, the peptidoglycans are found within the periplasmic space. Yes, are you getting that now? And I already told you that this region here is called your what? It's called your inner membrane, and I told you this upper side here is called your outer membrane. So by arrangement, you have outer membrane, you have periplasmic space, you have the inner membrane. Very good. Yes. Now, back here now, just leave this. This is the inner membrane. In this case, inner membrane. In this case, yes. So like I said earlier, all these things are peptidoglycans, the murines. And I told you that we're using red to represent NAM, and we're using the other one to represent NAC. Now, this is one thing I need you to know. If you look into this, you can see some strands here. We have one particular green strand here that links all the peptidoglycan. It moves and attaches to the what? To the inner membrane. So you can see it attached to the inner membrane. This particular component here that moves through the peptidoglycan layer and attach into the inner membrane, that guy is called lipoteichoic acid. Lipoteichoic acid. You can see where we bring that from now. So the lipoteichoic acid is responsible for what? For adherence. Is responsible for adherence because this remaining strand here can be used to anchor to a particular surface. So it is going to be the connecting branch between the inner membrane and the what? And the peptidoglycan layer, and it projects outward. You can see it projects outward. Good. Why these other guys here? This is the same for this. Why these other guys here? These ones here. Are you there now? Those ones are the teichoic acids. Those ones are the teichoic acids. They are the teichoic acid. So we have the teichoic acid. So we have teichoic acid and lipoteichoic acid. As you can see, the teichoic acid here is just responsible for holding the peptidoglycans to each other, holding peptidoglycans to each other. So the more compact the peptidoglycans are held together, the tougher the cell wall. So now that means we can easily say that teichoic acid is responsible for what? We can easily say that the teichoic acid is responsible for the toughness of the wall. Toughness of wall. I get it now, is what's responsible for the toughness of the cell wall because of the peptidoglycan. It holds all the peptidoglycan together. Now, each peptidoglycan actually is linked to each other by peptide bonds as well. They are cross-linked with peptide bonds. They are cross-linked with peptide bonds. You just need to remember that. They are cross-linked with peptide bonds to each other. So the teichoic acid is just giving it more stability. Same thing with lipoteichoic acid, is giving stability and adherence. So this is what I want you to remember. Teichoic acid basically functions to toughen the wall, while your lipoteichoic acid, aside from the fact that I've told you it will do adherence because of the fact it's connecting the membrane to outer side, I get me now. It will also help in latching. Latching there is just the same as adherence. Adherence, or it has adhesive characteristics. And when you say adhesive, that's the ability for it to attach to surfaces. Good. Now, because of that, this guy is considered to also be a virulent factor. Virulent factor. Because it's helping to attach. Anything that will contribute to attaching and all those things are virulent. While the teichoic acid plays more role when it comes to antigenicity. So you want to remember this, antigenicity. So teichoic acid will contribute to antigenicity. Antigenicity there is talking about the ability for it to express antigens. I guess me now, which are components that have ability to initiate immune responses. They recognize bodies and tend to stimulate immune responses. All right, so good. So we have antigenicity and lipoteichoic acid virulent factor. That means it can be able to trigger diseases. All right, good. So that's that for this one here. And when we come here now, you can see that the peptidoglycans are found within the periplasmic space, and you can see these green proteins here that are helping to attach the peptidoglycans to the outer membrane. They are called Brown's protein. They are called Brown's protein. They are called Brown's protein. B-R-A-U-N-S. So we have the Brown's protein. So basically, the Brown's protein, it binds covalently, because they can ask you, this Brown's protein, how does it bind? It binds covalently to what? To peptidoglycan. It will bind covalently to peptidoglycan. But when you talk about this upside here, it's not covalent bonding. I get it now. That up region is lipid soluble. So it just attaches itself to that to the outer membrane. as a result of solubility compatibility between them. So that's that for that. Now, this structure here that you see, which is still part of the outer membrane, is called the LPS. That's that lipopolysaccharide we talked about earlier, the LPS. And you see that there are strands from the LPS that move outward that form the outermost projections, and we call them the O antigen side chain. O antigen side chain. So we have the O antigen side chain. So these are the O antigen side chain. Now we have different kind of antigen expressions in bacterial cells. You have the K antigens, you have the O antigens, and you have the H antigens. The particular antigen expression you find on the flagella is the H antigen. The one you find on the capsules are the K antigen, while the one you find in the cell wall are the O antigen side chain. So just keep that in mind. All right, good and very good. So with that, we've been able to discuss the layout of the cell wall. Now, you know, I told you that you should be able to understand the staining technique that we use in differentiating between these two bacterial types. For instance, now, you can be told there that, oh, gram-positive bacteria, whenever they carry out gram staining technique, that they stain purple. Why this one stain red? There are reasons for those things, and you need to understand the technique behind it. So let's come here now. So we want to look at that gram staining. Gram staining. So it happens within 4 steps, and there are 3 major important components that you want to use in this process, in this technique. So you are going to be making use of something we call crystal violet stain. Crystal violet stain. So we have our crystal violet stain. You have your safranin. Which is also a dye. These are 2 dyes. Now you need your alcohol. Or we have the one they call alcohol acetone. You need it. And you also need the last one there, which is the mordant, and that's iodine. Precisely gram iodine. All right, good. So how does this staining technique work? On what principle does it work? Now, this is it. Peptidoglycans usually have the ability to retain primary dyes. Now, so you have to note that down. Peptidoglycans naturally have the ability to retain primary dyes. Why? So since gram-positive have thick peptidoglycan, that means they are more suitable to retain any dye that is put into them. Why gram-negative, now that has less amount of peptidoglycan, or some of them lack it, that means they won't be able to retain the dyes. Now, in your first step for your gram staining, they can ask you, what's the first thing you want to do during gram staining? First thing you want to do during gram staining is to add the primary dye first. So you want to put in the primary dye or stain with the primary dye. And what's the primary dye? Crystal violet. So you want to stain with CV, crystal violet, and crystal violet is purple in color. So when you stain, for example, you have a gram-positive bacteria and you have a gram-negative bacteria, when you stain the both of them with the crystal violet, I get what I'm saying now, they both appear purple color initially. So the both of them, they will both retain the initial purple coloration. Yes. Now, when they retain that initial purple coloration, we are now going to add on to them the mordants. The mordant, M-O-R-D-A-N-T. And what's the mordant? The mordant is iodine. That's the gram iodine. So when you add the mordant, the iodine there is going to help to allow the crystal violet stain stick properly to the cell wall of both the gram positive and gram negative. So we really want it to stain properly. We want the stain to stay and stick to the cell wall. I get what I'm saying now. So we want them to stick to tissues, and that's the work of any mordant. So in this case here now, the mordant we use here is iodine. So when you put iodine in, it will form something that we call the crystal violet iodine complex. Now, that means the iodine will merge with the crystal violet and help the crystal violet stick to the surface. That's the crystal violet iodine complex. Now, when that is done, the next thing that we'll do is a critical step. It's one of the most important steps here, and that's what? That's the addition of alcohol. So we are washing with alcohol. We'll wash you stained the gram negative and gram positive. Now you're not pouring alcohol on both of them. So alcohol or alcohol acetone addition. So So you're applying alcohol or alcohol acetone to the component there. And when you do that, what's going to happen is this. For the gram-positive, idea now, alcohol will cause a contraction. It's going to cause this peptidoglycan, instead of being loose, to be more compact. It's going to cause them to contract to themselves. And when they contract to themselves, imagine dyes have entered into these spaces here. If they contract, what happens? You are retaining the dye even much better. I get what I'm saying now. So when you add alcohol, it's contracting the dye. But in this case here now, there is less amount of peptidoglycan, right? So there is no way the dye can be retained. So when you pour the alcohol causes the crystal violet iodine complex that you had in both of them to wash out in gram gram negative. And when that happens, your gram negative will now start appearing colorless. Why? Because there is not enough peptidoglycan to have held together or retain the crystal violet iodine complex. But in this case here now, that the gram positive, you will start noticing that the purple coloration will appear. That it will still be retained. You know, it was purple already. It's still retained. Why? Because the alcohol could not wash away that crystal violet. Why? Why could it not wash away the crystal violet? Because it has caused contraction of the peptidoglycan, and the peptidoglycan now have They've retained, just like squeezing, they've retained the crystal violet iodine complex. Alright, so when that is done also, we have to counter stain. So they can ask you which of the following is the counter stain here. Is safranin. So we are going to be applying the counter stain. The counter stain, which is safranin. So when you add safranin to the both of them, what do you expect? Remember that peptidoglycan has contracted, right? And that contraction of the peptidoglycan is going to cause what? It's going to prevent the safranin from entering into the peptidoglycan even further. So you're not going to see the color of the safranin. Why? Because already there is no space, there is no pore for that to happen. But in this case here now, that is still loose, the peptidoglycan is still loose because you have less amount of peptidoglycan. When you put in safranin, it will retain the safranin coloration, which will make it appear red or pink. So what are we trying to say? During the gram staining technique, you have purple for gram positive due to retention of crystal violet. Then you have colorless, red or pink due to presence of safranin, that it absorbs the counter stain. So that's that when it comes to the cell wall of the bacteria. Now, on the other hand, plasma membrane, we don't need to waste more time on this one. The plasma membrane basically is responsible for transport. It allows the exit and entry of materials in and out of the cell of the bacteria. Do you get that now? Another thing it does that is very important is that it has the ability to initiate respiration at some regions. And what are the names of those regions? We call them the mesosome. How does this happen? Now we know that the bacteria does not have mitochondria, but yet it has its energy means of production. It has its means for producing energy. And how does it achieve that? It achieves that through what we call the electron transport chain. You can ask you this electron transport chain. So within the plasma membrane, we have the electron transport chain. I get now found in the mesosome, which is an infolding of the plasma membrane, and that enables generation of energy. Good. Then next we look at the cytoplasm. The cytoplasm helps in suspending all other organelles within the bacterial cell, like the ribosome, like your nucleoid, the DNA strands, and all those things. They are all found within the cytoplasm. So with what we've done right now, we've been able to conclude the morphology and structure of the bacterial cell. So do well to subscribe to our channel and also watch previous channel, as this some of the introductory concepts on these bacterial cells have been discussed earlier. Thank you.